Hello everyone, and welcome back to our chapter lectures. Today we will be covering the highlights or some of the main concepts from chapter 9, Patterns of Inheritance. As always, rely on the textbook as your primary study source, and then use these PowerPoints to help solidify main concepts as we go through. But there are some things in the text I don't cover here, so make sure you definitely use your book first and then rely on the PowerPoints second. The study of genetics is a fairly recent one, quote unquote. It's only really been in the last 200 years or so that this has become a discipline. So we are going to look through the discipline or the science of heredity, which is an old school term for genetics. So we can use these terms interchangeably. There are also going to be other terms that we can use interchangeably as well. And again, it just really has to deal with, is it the old school terminology or the more modern terminology? And I'll try to point that out as we go along. Heredity is the transmission of traits from one generation to the next. So genetics is the study of heredity, and the father of genetics is an Austrian monk named Gregor Mendel, and he developed this science in the mid-1800s. As he was working in his garden, he would crossbreed pea plants. And after doing that for several generations, he would notice that when he crossed two parents of differing traits, that would determine the traits of the offspring. So this was really a novel idea at the time, and it took many decades for him to finish his work, and a longer time after that for his work to become accepted by the public. And he would also come to develop sort of the standard in any sort of heredity or genetics study. So he would create purebred varieties where all the offspring are identical to the parent. Like, for example, he would breed pea plants with a purple flower with another pea plant with a purple flower, and their offspring would be purple flowered. He also developed hybrid varieties of these plants through his crossbreeding experiments, and that is where the offspring of two different purebred varieties, So he would then crossbreed a purple flowered plant with a white flowered plant, and then those offspring were no longer purely bred, they were called hybrids. Another analogy that probably would be more familiar to you than plants would be dog breeding. So we all are familiar with what a purebred dog would be. For example, a poodle. That poodle's parents were poodles, the grandparents were poodles, as far back as you can go. It's only poodle DNA in that dog, as opposed to, let's say, a mutt, quote unquote, would be, let's say, a poodle bred with a beagle. Okay, those resulting puppies would no longer be purely bred. They would be hybrids, or if we're talking about dogs, a more general term would be a mutt. And here's the setup for his experiment. So he would breed these plants himself to make sure he knew what was being fertilized and what was being crossbred, etc. He would take the pollen off of the stamen of a white flower, for example, and the pollen is the male part or the male gametophyte of a flower, so it has sperm in it. And then he would then dust the carpel or the female sex organ of a purple flower. So then the sperm and that pollen would eventually dig its way down and fertilize the eggs down here at the base of the carpel. And we all know what happens when sperm fertilizes an egg. Little babies are formed, and that is the resulting pea pod. So the pod is just the ovary of that flower, and the little tiny babies, little baby plants, are the peas. And so then he would go ahead and plant those seeds with the little baby plants in them, and then he would see what grew. And that was his basic setup for what is now called a genetic cross. And in terms of crosses, it's literally just breeding different varieties with each other. It's what we mean by a cross. And we have some new terminology with this image. So as we study these genetic crosses, we simplify the generations to letters and numbers. So the parents, all right, this initial purple and white flower, we call them the parental generation. And then we shorthand that to the letter P. So the P generation is the parents. And the children, or the offspring of the parents, we now can shorthand to F1. The F stands for filial. It's Latin for family. So F1 would be the first generation of the family, quote unquote, or the children of the parents, or the offspring, etc. And then if we take it even further, let's say we were to crossbreed two F1 generation plants with one another, their resulting offspring, or their resulting children, we would then denote as the F2, or that filial generation 2. And that would be either the grandchildren of the P generation or the children of the F1 generation. So those are shorthand terms, abbreviations you will definitely need to know and be familiar with for this chapter, and understand what's coming next. So imagine you have two purebred dogs, let's say a poodle, for example, one male and one female. Use the correct genetic names to describe the next two possible generations. So I'll give you a couple of seconds. You can pause this now and do it yourself. Say it out loud or write it down. And then we'll go over it in a moment just to make sure you have that clear. Okay. So our two purebred dogs that we start with, who are doing the initial breeding, those will be our parental generation, or the P generation. Then their puppies would be the F1 generation, so the offspring of the parents is our F1. And then if we bred those puppies to one another, which for ethical reasons we will not do, but hypothetically, those resulting puppies would then be the F2. So hopefully that makes a little bit of sense, and it also will help if you do this with your own family. So your grandparents would be the P generation, your parents would be F1, and you would be F2. Or your parents are the P generation, you are F1, and then your children are F2, so on and so forth, and it just goes on and on and on. As Mendel was experimenting with his crossbreeding and his genetics, his heredity studies, he discovered that there are certain laws to heredity. So it wasn't just random as was thought before. So the prevailing theory of the day at his time was that you were completely unique to anyone and anything, and it wasn't well understood. Actually, it wasn't understood at all that your parents actually passed down their traits to you. The current thinking of the day was that you were just amalgamated, sort of just randomly. Your essence was your own, if that makes sense. But this was really the first scientific study showing that no, in fact, we get our traits. We are indeed different versions, mixed and mashed, of what our parents passed down from them. Okay, so these are things that he discovered and then later published that really was the framework for the discipline of genetics that we still study today. So he discovered that there are alternate versions of genes, and in his day he coined the term gene as character. So Molecular biology hadn't been invented yet, so genes is a more modern version for a character, and those terms can be used interchangeably throughout the chapter and this lecture. So alternate versions of genes that account for variations in inheritance. And Mendel coined the term traits in terms of inheritance. So those terms can also be used interchangeably. His genetic crosses led him to develop 4 hypotheses about inheritance. So definitely make sure you can recall these 4 by memory for any future quiz or exam. And this is really what he discovered throughout his decades of crossbreeding his plants. For each gene, an organism inherits 2 versions, and we call those 2 versions alleles, one from mom and one from dad. So we've actually already learned a little bit about this when we were covering meiosis, when we looked at how the homologous chromosomes paired up. Well, those alternate versions, those alleles, are carried on homologous chromosomes in all of our cells, one from mom and one from dad. The alleles of a gene can be the same, and we would call that homozygous. Again, the prefix homo is the same, or different, heterozygous, hetero being the prefix for different. If an organism is heterozygous, meaning there are different versions of each trait carried on those homologous chromosomes from mom and dad, then the law of dominance prevails. So that dominant gene, which we would call an allele, that dominant allele is what determines your appearance, and the recessive allele is masked. So if we're talking about, let's say the gene for eye color, for example, your mom gives you her version and your dad gives you his version. So those 2 versions would be alleles. And let's say mom gave you a blue gene, dad gave you a brown gene. So you would be heterozygous for the gene for eye color. And in terms of eye color, it's much more complicated than Mendel's laws, but for argument's sake, let's just say that the brown allele is dominant to the blue, meaning if you have one brown allele and one blue allele, that brown allele is going to dominate or overshadow that blue allele. That's what it means to be dominant versus recessive. So if you have that genetic makeup, you're going to have brown eyes, even though you technically do have a gene for blue. That brown is going to mask or overpower that blue gene. The reason for this, so you may be thinking, well, why? What makes one allele recessive and one dominant? Not exactly sure, but there is a theory, and then some research is going into this now currently, to suggest that a recessive gene or a stretch of DNA, which is what a gene is, the resulting protein made from that gene is broken or fractured. So there's something about the the protein structure coded for by that gene that is not as strong than a dominant gene. And then the last law, sperm and egg carry one allele for each gene. Of course, there's mom and dad's complement, and then we can also see that because upon fertilization of our own selves, dad's sperm and mom's egg created us, and those each carried one version of every gene to make a human. So Mendel basically put out there to the scientific community these 4 laws. And a lot of this we've actually technically already covered when we went over meiosis, so maybe do a recap of that. And also, a lot of the genes talk we covered is covered in chapter 10. And we actually don't cover that in this class, but if you want to give it a read, it will definitely help sort of make sense of when I say genes make proteins and genes are stretches of DNA, etc. Okay, so then let's put this all together, what we've gone through so far. So, of course, in genetics, each gene is represented by a letter, and it's just shorthand because we like to simplify things. Okay, so in this example, we are just looking at the gene for flower color, purple or white, and we're just going to pick a letter. There's no rhyme or reason, there's nothing assigned to anything else. It'll already be picked for you, like on any quiz or exam or in your book. It's just whatever the teacher decides. So in this example, we're going to shorthand, or we're going to abbreviate the gene for flower color to the letter P. So let's look at our parents, or our P generation. We are crossbreeding two purebred flowers. And the reason why we can tell that they're purely bred is because there's only one type of gene, one type of allele in the whole makeup. So we have a purebred purple flower denoted by an uppercase P and another uppercase P. Again, the reason why there are two of these alleles is because this flower got one from mom and one from dad. So now there's two. Same thing with our white flower. So we have a lowercase p and a lowercase p. Anytime we see an uppercase letter for a gene, that is going to be our dominant allele. So purple, we can already see, is going to be dominant to white, and we denote that by making the purple color a capital P and the white color an undercase P. So then let's crossbreed these two purebred flowers. So the law of segregation, again, which is when our homologous chromosomes separate in meiosis 1, we have one P going into one egg, another P going into another egg. So they separate or segregate. That's the law of segregation. So that all the eggs made by our purple flower only contain one possibility, uppercase P, because it's a purebred. Same thing with our white flower. We have law of segregation, where our homologous chromosomes separate in meiosis 1 to make sperm or egg. And so we have one little P going into one sperm, another little P in another, so that all the sperm this flower can carry are all little P. There's only one possibility, again, because these are purebred plants. So then we have fertilization happening. This is where sperm and egg fuse to then give us our children, or we now call them F1 generation. Also note that the F1 generation of purebred parents, two different purebreds, are always going to be hybrids. So we're no longer purely bred, but they're all purple. Okay, so again, that sort of lends to the fact that that purple color is dominant to white. So we see the genetic makeup of our F1 generation is going to be big P, little p. That means that we are heterozygous, so we have the two different allele versions, and purple is dominant to white because purple is the only color that's showing forth. So now let's crossbreed two F1 hybrids with one another. So then two F1s, we undergo the law of segregation again. So we separate our alleles, and we have some eggs are going to carry a big P, and now some eggs are going to carry a little p. So there's a little variation here because we're hybrids. And that is where we can use Punnett squares to really show our F2. Okay, and a Punnett square is what's here down at the bottom. And you may have done some of these in high school. They're fairly common, but if you haven't, basically our setup, you perform the law of segregation. So you separate your alleles. And then for parent 1, you just put your two alleles on this axis. Parent 2, you put the two alleles on this axis. And then you literally just drag and drop. So big P is going to be this possibility for child. Little p, big P, so drag that over. We have this possibility. And then same thing down here. We got big P, and then little p, little p. And we can see in our F2, we actually have a lot of variety in the genetic makeup, which we now call genotype. which is literally just the allele combinations. Is it big P? Is it little p? And we also have some variation in how the flowers look or their appearance, which is what we call a phenotype. And from these Punnett squares, we can get ratios. And as long as you can count, you can do ratios. So if I were to ask you, what is the genetic makeup of our F2 generation? All you would have to do is tell me how many of each kind of allele combination there are. We only have one big P, so then we just write that here. And we've got two that are big P, little p. So then we put that. And then we have one little p. So our genotypic ratio is 1 to 2 to 1. Phenotype is how they look to our eyes. So we have 3 purple and 1 white. So our phenotypic ratio would be a 3 to 1. So that is taking you through everything we've learned so far, plus introducing the Punnett square, which if we're just doing one trait at a time, we call it a monohybrid cross. Introducing you to the terms genotype and phenotype, and showing you how to do the ratios for each. Okay, so again, for your notes, or if you do flashcards for definition, which I always recommend, phenotype is an organism's observable traits, like in terms of flower color, is it purple or white? And genotype is an organism's the genetic makeup, or the allele combination, like is it big P, big P, little p, or little p, little p. And then again, I went through how to, I told you how to get the ratios for how they look and the ratios for their genetic makeup. And you literally just count and you group like with like. And also keep in mind, genes are located on chromosomes. So think back to chapter 8, meiosis. Those homologous chromosomes that line up and then separate when we make egg and sperm. Remember, a chromosome is just a long molecule of DNA and protein. And that DNA serves to give us the genes, which will eventually become proteins. And then proteins are everything in the cell. How the cell behaves, the cellular machinery, what we eventually look like, how our organs function. Every single thing about us basically is a manifestation of some type of protein or program within the DNA itself. And so here we can see how a trait becomes homozygous or heterozygous, and how an organism becomes homozygous or heterozygous. If you are homozygous dominant for a trait, let's say eye color, for example, you're homozygous dominant. That means that your mom had to give you the dominant version of that gene, and your dad gave you the dominant version. So they gave you the same version. So they both gave you a big P. So you're homozygous dominant. If they both gave you the recessive version of that trait or gene, then you're homozygous recessive. So you're the same, but it's the recessive version of the gene. And then if they gave you one of each, let's say mom gave you a dominant version and dad gave you a recessive, then you would be heterozygous. And we don't say heterozygous dominant because it's just de facto. If you're heterozygous, the dominant is always going to show through. So we can just say heterozygous for that one. And it's just assumed, of course, because there's already a dominant gene there. So here's a good time to pause this and try to answer these on your own, and then I'll go over the answers here in a moment. So if you want to pause this and try to answer and then resume it in a couple of seconds, we can go through the right answers for these. So genes come in different versions called alleles. What term describes the condition where the two copies are identical? Homozygous. What term describes the condition where two copies are different? Heterozygous. All right, if two plants have the same genotype, must they have the same phenotype? So I would say yes, but if two plants have the same phenotype, must they have the same genotype? So that one is a no. So remember, let's use our flower color example. If you have a purple flower, you can either be homozygous dominant, so big P, or you can also be heterozygous. You can have a big P and a little p. So you would have the same phenotype or the same look, same color, but a different genotype or a different allele makeup. If two plants have the same genotype, must they have the same we just went through this. Same genotype, same phenotype. Again, I'd say yes. Okay. So everything we looked at so far, okay, basically covered Mendel's law of segregation. And again, go back to the meiosis chapter. All the law of segregation describes is when those homologous chromosomes separate during meiosis and one copy goes into an egg, or goes into one egg, another copy goes into another egg or sperm, whether you're male or female. This is just where the homologous chromosomes separate when we make egg or sperm. And then we can see the resulting match up from that if we do a Punnett square. Something else interesting that happens during meiosis that we have already covered in our class, but it's really attributed to Mendel's discovery of this, which we're going to relearn, is that our homologous chromosomes independently assort, meaning they line up randomly on the metaphase plate during meiosis 1. And because of that random assortment, they just sort of line up however they wish, that gives us a plethora of genetic variability in the resulting egg and sperm. So the Punnett squares crossing one trait at a time, again, are the monohybrid crosses, mono for one, just one trait, and those are easy. But in this class, we're going to take it up a notch. So we're actually going to cross two traits at once, creating our dihybrid cross. Di is the prefix for two. For these crosses, you'll always be given the information regarding dominance and recessiveness, heterozygous, homozygous, etc. So all of the background information will always be given to you already. And then you just basically work out the law of segregation and the law of independent assortment. So basically, you are putting meiosis into action, and we're going to see what resulting egg and sperm, and then what the resulting offspring are going to look like, genetically speaking. So Mendel crossed homozygous plants, or purebreds, with yellow round seeds. So here we can see the genotype for a plant that has yellow round seeds. So the R is going to be the letter for seed shape, round or wrinkled, and we just picked the letter R. And then the letter Y is going to be for the seed color, and we just picked Y for that letter for seed color. So a purely bred plant with yellow round seeds is going to have the genotype big R, and big Y. Again, why are there two big R's? Well, because there are always two versions for every gene. And this is one gene, and this is the gene for seed shape. Same thing for the letter Y. Why are there two Y's? Well, because there's always two alleles for every gene, one from mom, one from dad. And because we're doing two traits at once, we put those together. So we've got big R, big Y. So we're crossing that purebred with another purebred, but a homozygous recessive plant. So this plant is green and wrinkled seeds. So because green is recessive to yellow, we're doing 2 lowercase r's. And because wrinkled seeds are recessive to round, we're doing 2 little y's. You always keep the letters the same, but you just denote whether they're dominant or recessive by capitalizing or undercasing. So we wanted to know if the 2 characters from each parent were inherited as a package or if each character inherited independently. And just a little preview, a little hint, it's going to be they are inherited independently, hence the law of independent assortment, but we'll actually see how we can be sure of that. So we answered this question by crossing some F1 hybrids. So let's take a look at this. Here are the P generation. Here are the parents, our 2 purebreds we just went over. Big R, big Y, big R, big R, big Y, crossed with little r, little y, little y. Okay? Again, the only gene combination that this parent can give is big R, big Y. All right? So it just gave you one of each possibility from each gene. Same thing with our other parent, little r, little y. That's all the genetic makeup is. That's all that's there, so that's all they can pass on. But we're going to look at what happens when we cross F1 hybrids. So the resulting F1 generation, we just literally combine these 2 egg and sperm. So we've got 1 big R and 1 little r, 1 big Y and 1 little y. And that's just looking at what was there. Okay, we just sort of put them together, and that's how we get the genotype for the F1. But when we cross F1 hybrids with one another, big R, big Y, we get a setup that looks like this. And the reason why our F2 hybrid generation, okay, our F2 generation looks like the chart on the right as opposed to the chart on the left is because each one of these alleles independently assorts with the other. So for example, this is called the FOIL method. And you can do this on your own or just in your head, but in our first square here for parent number 1, we will combine the first allele of the first gene with the first allele of the second. So that would be a big R with a big Y. That's what we have here. And then for our second square, we combine our big R with the little y. And that would be what's here. And then we go to our second R. We independently assort it with the big Y. That's what's here. And then we assort the little r to the little y. And that's what's here. Basically, we're just giving every combination possibility. Okay? And then because we are crossbreeding F1 hybrids, which are all the same, we have the same setup on this axis as well. So that's how you get the setup for a dihybrid cross, the FOIL method. And if you wanted to go onto YouTube and look at the FOIL method in more detail and how to set these up, that would probably be very helpful. But that's the easiest way that I can tell you how to do it. And then, of course, from there, you just drag and drop every single square, and that gives us our genotype and phenotype. So then, after we've done this cross all the way through for our F2 generation, we give our ratios, okay? And now, if we're doing a dihybrid cross, you generally will not be asked to do genotypic ratio, because that would be a very long ratio. But you will be asked to do phenotypic ratio. So how would we get the phenotypic ratio for our F2 generation? Well, you literally just group like with like and then count how many there are. So there are 9 seeds that are yellow and round. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we'll put a number 9. What about green and round? 1, 2, 3. What about wrinkled and yellow? 1, 2, 3. And then what about wrinkled and green? There's just 1. Now you put all those numbers together in descending order. So the phenotypic ratio for our F2 generation would be 9 to 3 to 3 to 1. And FYI, anytime you cross a heterozygous F1 hybrid with the same, so 2 that are identical and heterozygous, you always get this 9 to 3 to 3 to 1 ratio. So if that question ever comes up on an exam and you have crossed 2 F1 hybrids, then you don't even really need to count because it's automatically a 9 to 3 to 3 to 1. So that takes us through classical genetics, or we can also term it Mendelian inheritance, because Mendel was the guy who sort of discovered all of this and actually played out these crosses over decades and got tons of data to support what he was publishing. So it's well established that everything we've seen so far does happen, and we can see it happening, and there's really no dispute. Except when there are always exceptions to the rule. And so we're going to learn more modern genetics, or variation on Mendel's law. You can also call this non-Mendelian genetics. So sometimes, genetics is much more complicated than the law of segregation and independent assortment, the result that Mendel saw. Remember, Mendel lived in the mid-1800s, so modern molecular advancements had definitely not been thought of. He had no idea what DNA was. He didn't know the mechanisms for what he was seeing. He was just describing what he was seeing. Today, we do have molecular biology. We know what DNA is. We know what genes are. We know what proteins are. And we can also determine that heredity is much, much more complicated than even what Mendel was describing. So we're going to look at some examples on variations of Mendel's laws. 4 variations that Mendel didn't know about, he hadn't discovered yet, and if he had, he wouldn't really have had the capacity to explain what he was seeing, again, because molecular genetics definitely a long way off. So the first example is something called incomplete dominance, and it's pretty much exactly what it sounds like. If a gene is incompletely dominant, that means that your F1 hybrids are going to be a blend of the 2 parents. So for example, if we have 2 purebred flowers, all right, a red and a white, red is going to be dominant to white. The resulting F1 generation is going to be big R, little r. But instead of our F1s being red, right, because of this dominant allele, both of these characters blend together to give us a mix of red and white, which is pink. So that dominant allele is sort of incompletely masking the recessive one, if you want to think about it like that. And you can see that nothing new has been created. This is just different manifestation of this genotype, because when you cross 2 F1s to give an F2, you can see that homozygous dominant and then homozygous recessive are red and white, and it is only the hybrid of this gene that shows that blended characteristic or that blended phenotype. The second variation on Mendel's law is called codominance, or it's also termed multiple alleles, either one. So codominance occurs when a gene has more than one allele. So this is definitely going against Mendel's established 2 alleles per gene theory. And a really good example of codominance is our blood typing. So blood type is known as the A, B, O blood system. And it's actually determined by 3 alleles, A, B, and or O, and giving us 4 possible phenotypes. So blood type A, blood type B, blood type AB, or blood type O. And if you don't know your blood type, that's a really good thing to just know about yourself. So find out what your blood type is and just make a note of it just in case anything comes up in the future. But the combination of the, you can either be homozygous dominant for the A blood type, you can be heterozygous for the A blood type, same thing with the B. You can have one of each. So if you're blood type AB, you've got one dominant A and one dominant B. Or if you're blood type O, you have 2 copies of this recessive, or this O allele, essentially is what it can be equated to. And all that means is, in terms of your A, B, or O, it just has to do with what type of carbohydrate is on your blood cell, on the surface of your blood cells. Is it carbohydrate A, carbohydrate B? Is it both of them? Or if you're blood type O, you don't have any on the outside. Okay, number 3 for the exceptions to Mendel's rules is called pleiotropy. And this is when one gene determines several characteristics. So an example of this would be sickle cell anemia. In order to have sickle cell anemia, you have to be homozygous for that gene. So each parent has to give you a copy of the sickle cell gene. And if you inherit both copies, you have sickle cell disease, which basically causes your blood cells to become misfolded or sickle shaped like this. And from that, a whole host of problems arises that can determine several characteristics, like you don't get enough oxygen to the body. There are other systemic issues like weakness, pain, organ damage, paralysis. So you can see that one gene gives you many, many different characteristics. And pretty much the opposite of pleiotropy is going to be polygenic inheritance. This is where 2 or more genes give you a single character. So these are, the last 2 here are just opposites of one another. So a good example of polygenic inheritance would be skin color. There aren't just 2 or 3 skin colors. It's a wide spectrum of skin colors. Everyone's skin is a little bit different. And again, that is because skin color is a polygenic trait, meaning your skin color is determined by many, many, many different genes. Okay, so all those different genes come together to give you one phenotype. And then, of course,